So I thank the very few of you who came to this talk. You're going to hear a wonderful talk where we aim to predict the determinants of the emergence of the 2009 flu. So the original work, which was published, included mostly computations by Daphna May-Rose from my lab with a lot of help by Tomer Hertz. And I'll present some of that, but since then, and there were minor, like preliminary experiments by Richard Webby, but since then, Richard did a lot of more experiments, making it really, really cool project, and he was kind enough also to give me some of his slides, which are much superior to anything I've ever used. So you'll be having a wonderful experience here. So influenza A is responsible for seasonal epidemics and at times major pandemics. It is subtyped by two surface glycoproteins, which are going to be the heroes of this talk. So the main character is going to be the hemagglutinin, which facilitates binding of the virus to the host cell, and neuraminidase, which essentially facilitates the release of the budding virus from the cell. We've experienced major pandemics, including the Spanish flu, etc., and including the swine origin flu that I'll be focusing on. And this slide is actually making the point in the talk in that the swine flu didn't live up to the expectations in terms of death toll, but it was very unique in that it managed to invade the human population really, really quickly, suggesting that there is something unique there, and that's what we tried to find. So this slide gives some of the histories there. So the influenza genome includes 8 genes. It is based on, and the new pandemic was based on the so-called a mixture of two strains. So there is a North American parent, which donated 6 genes, and the Euro-Asian parent from both are in swine, and they donate 2 genes, the neuraminidase and the matrix protein. And this is how people used to measure, to model influenza in animal models rather than human. So ferret is considered a good model for years because the symptoms are very similar to human and for other reasons that I will not get into. And the very common apparatus used is something like that, where you have ferrets both in the same cage, and there you test for direct transmission, and also in a different cage, which is separated, and that checks essentially the ability of the ferrets to sneeze and transmit to each other, and that's important for pandemic, as you could imagine. Now, and this is result from Richard. So, and again, this is this apparatus, and you can see that you infect the red animal, and it transmits both to the near neighbor from the same cage, but also to the remote animal in another cage. So that was an important property of or characteristic of the pandemic. Now, this slide shows you that this is unique because neither parents, neither the North American nor the Euro-Asian were able to do that. And the immediate question is whether reassortment, this process of combining the genome, is enough, and I'll show later that it's not. So there's something else going on there. And it brings us to the barrier between, for animal to human transfer, which is based both on the exposure to infected host, of course, but also to the population immunity and the receptor preferences. I'll touch these 2 aspects, and we'll start with receptor preferences. So the host receptor is a sialic acid, and it should be in the upper respiratory tract. And And there are two chemically distinct types of receptors. One of them is marked as alpha 2,3, and the other is alpha 2,6. Now, birds have in their upper tract the first type, and human are mostly of the second type. And the pig has both, making it a good channel to transmit between them, and also a good incubatory place for the resource of itself for producing pandemics, and that's actually what happened this time. So the thing that binds to the receptor is the hemagglutinin protein shown here. It's a homotrimeric elongated protein. And you see here one subunit with binding to the sialic acid receptor. This region here is called the receptor binding domain, and we will focus on this region. So it's obvious that we should look at that because that's where the binding is, and we want to look for specificity. So the computational goal is described here. So the idea is to identify amino acid positions in the hemagglutinin that appear to discriminate between the novel H1N1 type from isolates from other groups. And the specific aims would be to find the unique properties of the pandemic versus the seasonal variants. And also, in another thing, to discriminate between the pandemic versus groups that are in the swine itself. And the means here is sequence analysis, very, very simple sequence analysis based on supervised learning using alternative decision trees. And that's a quick slide that shows what we have. I'm not going to go over all the algorithm, but it's based on, so we have a set of sequences marked here as xi, and these would be hemagglutinin sequences. And the important thing is that they will have a binary annotation, which is marked here as yi, and that would be pandemic versus seasonal. And the algorithm goes over the sequences and try to segregate them based on the amino acid positions in the sequence. And the idea is first to find a good segregation, because if we cannot segregate them, then there's nothing to proceed. But what we actually aim for is to find the amino acid positions that are responsible for the decision. Now, we want to get a small number of these, so we derived a tentative ranking of the results. So we just multiply the number of appearance of each amino acid position by the mean of detection iteration and the maximum of the prediction score of the sequence. And that seemed to be okay. And of course, the larger the contribution is, the more important the position is. So it's all based on sequence databases. So Daphna collected sequences from the NCBI influenza database, and she made two sets. One is for and each set included hundreds of sequences. So one of them was for pandemic hemagglutinin versus seasonal, and the other was for pandemic versus swine. And then she got rid of redundancy, aligned the sequences, and focused on the receptor binding domain, and just plugged it into this supervised AdaBoost trees, and that's what she got. So the result here, you see that this is a precision, and you see that there's very good separation here, right? We are over 90% quality. And in this case, with the pandemic versus seasonal, it's all based on 10 positions. And in the other case, with pandemic versus swine, it's all based on 13 positions. So we looked at each position specifically, but I'm not going to bore you with that. Here is the overall picture of pandemic versus seasonal. And what you see here is that for the most part, the detected positions are either directly on antigenic site or very near antigenic site. And that makes sense because they could change the stereochemistry of the sites, preventing the amino acids from detecting or making it difficult for the for the immune system to detect these positions. And that's in line with the fact that the human population was not immune for this particular strain. Now, and that's the most interesting result. So the virus made its way from the swine to the to us, and the question is why. So the 13 positions that are responsible for that, or appear to be responsible for that, 10 out of them are detected near or in the binding site. So that also makes sense, that the sialic acid binding site, so that can explain how the specificity changed. And from now on, it's essentially only experiments. So Richard did a lot of experiments. Some of them were presented in the original publication, but those are relatively the boring ones. I'll presenting new data. And he looked at many positions, but I'll be presenting only results for the firstly ranked position. That's a very minor substitution, arginine 149 to a lysine. And I'll show you that it does tremendous. So this is a North American parent. And again, we have this apparatus, and you see that the parent can transfect the neighbor, the green neighbor, but it cannot transfect the animal in the other cage. And also, and the transfection is not very large, but only when there is a mutation, and this one position mutation, that increases the transmission really strongly. So it's 100% transmission, actually. So the question is, what's happening there? And for that, Richard analyzed samples from various tissues. And this is the original, and this is the mutant. And the only difference here is in the nasal turbinate, which is in the end of the nose of the animal. And you see here that the mutant was found there more than the normal. This slide shows that the binding was higher. So he used a marker to mark the virus. This is just a control, and you see that with the parent there is almost no binding, whereas in the mutant there is much more binding. Here with more yellow. The point here is that there is more yellow, which is the virus. The blue is just the nuclei, a marker of the nuclei showing where it should bind. This is a glycan-erase assay showing that the difference is not due to change in specificity. This is the parent and this is the mutant, and basically you see that the profiles are similar, so it doesn't matter exactly what it is, but that's not the question. So what it is really, and this slide shows that it's due to changes in increased avidity. So the stripes results are controls, and the green is the parent, and you see that it has a certain avidity. But the red, which is a mutant, has a much stronger avidity. And recall, this is just a single mutation. So what could a single mutation do? We were curious about computationally. And for that, Turkan Haliloglu did normal mode analysis to find the most important modes of motion of the protein. And this residue, 149, was responsible for, was found as a major hinge for the motion. And here what you see in this slide is the correlation between the motion involved in this residue and other motions in other parts of the protein. And you see in red here, like that's the scale bar for the correlation. And in red, you see that it correlates with and controls the motion in the antigenic sites and also in residues that are in direct contact with the sialic acid receptor. So it seems to control the motion, and that could explain the importance of this residue and why a minor substitution such as arginine to lysine can cause so much difference in the phenotype. Now, this is the coolest result of all, and it explores the interplay between the neuraminidase and the matrix protein, those that are exported from the other parent, and the hemagglutinin mutant. as I told you, the reassortant virus here can transmit with direct contact really greatly. You can see that it affects the green animal, but it cannot go transpass to the other cage, so it cannot sneeze out. But the mutant can do that too. You see the blue lines here. So now that we know that neuraminidase is involved, the question is, what does it do? And this slide shows that it's just more active. It can facilitate the release of the virus from the host cell after budding quicker. So the green and red are the parent. The blue is the original pandemic, and the red is the mutant with the new neuraminidase. And that's the take-home message. So what do we need for transmission? So we have the infected ferret, and the virus has to propagate to the end of the nose. And for that, we need AJ1, which is we need the virus with more avid hemagglutinin, which seems to be the case with the mutant. And then this thing has to be detached for transmission with the sneeze. And for that, we need also a more active neuraminidase for this part. So that's the summary of what I showed from the experimental point of view. And now to the conclusion. So it's a good example of where computation saved a lot of experimental effort, because before this kind of approach, people used to spend a lot of time and money on using random mutagenesis to detect the amino acids that are responsible for transmission. And there was success, but the computation seems to save a lot of time here. And I should emphasize that the computational method is general and maybe applicable for other cases, and it's available. And then I want to acknowledge the people who did the work. So Daphna Meiroz and Noam Chen in my lab, Turkan Haliloglu from Bogazici University in Istanbul, Tomer Hertz. Well, you can see them all. And of course, Richard, who did the really interesting part of the work. I'd be happy to answer questions. That we look at the binding site and analyze it structurally, or?
Essentially, you save time by computational effort.
Right.
You develop a machine learning device, but if you could just have looked at the binding site, you would have missed 3 or 4 residues, but overall at the end, were they important?
Yeah, actually, so first, not all the residues in the binding site were important, but here you see this residue? And this is a binding site. So this residue is actually far from the binding site, and nevertheless it's so important. It scored the highest, and you see, so.
Okay, I have a short question. Have you checked also phylogenetic inference, because this is a standard tool to check what are the difference between virus strains, especially in influenza, and how great was the overlap to your sites you found?
Right. So we compared the computations to various other approaches, including phylogenetic. So I mean, you see of course, you can segregate the sequences based on the phylogeny, but we at least in our hands, we failed to infer back the differences in the sequence from the phylogenetic tree. We also used other methods based on mutual information and the like. And in fact, I think this residue came out also in these methods, but other residues did not.
Okay, thank you.